Okay, coming to the next talk. That is my talk I give to you. And the theme of my presentation would be how does formulation development triggers device development in the end. Yeah. And I would like to give an overview what kind of parameters are influencing the selection of a potential device later on. That could be a little bit more than someone expects. Yeah. And this presentation is focusing on biological drug development, not on NCE. Yeah, that could be a little bit different. But for that, you need a holistic approach from the early beginning to the later device and all the decisions you have to take over the development time. First of all, I would like to give a bit of a background about Boehringer Ingelheim, where we are and what we are doing and why we are doing that. Yeah. And then we could go to the drug product development and to options for devices. Okay. Good. Just a quick slide here that you can see that the market is still growing for biotherapeutic products in 2018, and BI also invest a lot of money to increase the capabilities for manufacturing and research of new molecule entities. And Boehringer Ingelheim is a family-owned company which is founded in 1885 in Ingelheim, Germany, and is focusing on three business units like human pharmaceuticals, animal health, and biopharmaceutical contract manufacturing. In 2018, around 50,000 employees worldwide are working for BI, and BI spends R&D expenditure of around 3.2 billion euros. On this slide, you can see the global research and development presence from Boehringer in Europe, North America, and Asia. I would like to point out that the R&D sites in Ridgefield, US, Biberach in Germany, and Vienna in Austria are parts of the NBE development from these sites that you can see here. And we have extensive capabilities for biological drug manufacturing, and with research beyond borders, BI harnesses emerging science for and beyond our own therapeutic areas. Okay, coming to the really thing here about potential antibodies and molecules. We can see here illustrated which proteins Boehringer is dealing, and in the last years there was a huge change to more and more complex molecules related to research, stability in drug product, to have a higher efficiency and to help patients. And this ends in a higher variability of the molecule formats, and this makes standard solutions for drug product development, especially formulation development, not at every time possible. And all molecules can have a different behavior and requires in most cases an individual composition of the formulation. And not all formats and all data you gain during the development makes a prediction really helpful. You have to do it once more and once more due to the complex molecules here. Just a short slide to the drug product development for biologicals. As I mentioned before, drug product development needs a holistic approach. You have to think from the beginning to the end. And it starts with the drug substance manufacturing in the end. We have here the formulation, which have to deal with different requirements and properties of the molecule. Then you have the primary packaging with all the input parameters like impurities, forces, and all these stuff related to the compatibility of the drug. You have the filling process, which can influence the product quality of the antibody later on. And after that, you have the device, and you have to take the decision what kind of device you would like to use in the future. It's more an ongoing discussion during the development. On this slide here, you can see that the scientist, which is responsible for the development, has to think about, has to have the end of the thing in mind later on. This means that when we compare it to a chess match, you have to think on every move you have to take, you have to evaluate it according to their effects. And that is really necessary to know that you have in mind some clinical and marketing needs. You have in mind the primary packaging, the combination product with the device, the API properties, and you have to think about all these stuff during the development, because the chemical molecules can influence the stability, and physical parameters can change the selection of the later device. Coming back to what could be helpful to have later on when we thinking about a 1 ml device or an injection less than 2 ml, that could be that we need more flexible in formulation development, and in that case it could be helpful to have a higher concentration within the formulation of the protein. And what does it mean in the end? For many therapeutic indications, especially subcutaneous injection, there are some high concentrated liquid formulations needed. It could be based on market needs, competitiveness, patient compliance, administration, self-administration. There are different reasons why we're talking about an HCLF development. And especially for antibodies, high doses are required, and this means that a higher concentration of formulation is needed later on, and you have to keep in mind that all the molecule properties can influence the stability. Higher the concentration will be. And later on, when you think about physical parameters like viscosity and a higher concentration of the protein, this can influence the device selection as well regarding needle diameters or forces which is applied with the auto-injector. Or it could end in that case if this patient is not able really to have a manual injection later on. What is really a high concentration? That is really a long story, I think. It could be 50 mg or 100 mg. Sometimes we're talking about 500 mg per ml. When we have 1,000 mg per ml, that is more a pill, and that is a different application. But that are questions we are facing for, and how we can select during the development the right concentration. And we could not take it on an isolated base. That's a formulation development guy think, okay, that it's possible, I do it on a highest way. And later on, the device guy said, oh my God, I couldn't get into the patient on a suitable way. It ain't a lot of pain or whatever. Molecule properties have a strong impact. As I mentioned before a couple of times, it could be a DNA sequence, primary amino structure, valences, thermal stability, hydrophobicity. All these molecule properties can have an impact on the stability, but you can handle that different behaviors during the formulation development to have different compositions, put it on stability, and found out what is really the suitable formulation later on. But when we're talking about excipients in the formulation, all these excipients can change the physical parameters as well. Like viscosity. Sometimes molecules are don't like shear stress when you put them through the needle, and higher the concentration is, you have to put more pressure on it, and you have to bring more molecules to through the needle, so that can impact some shear stress and the protein quality. And for that, it is really helpful to have at first for an HCLF development that you have an integrated testing for potential higher concentration, and you have to consider the device-related parameters from the early beginning. You can gain experience with the molecule within the transition phase from research to development, to have first spiking studies or short-term or stress stabilities to show how does a molecule behaves. And then you can plan during the development with additional studies what should be the right formulation and the right device later on. That is really, really needed. Coming back to the stability and the challenges for HCLF development, when we are talking about drug substance processes, when the BDS is ready, you have product quality data, it's a snapshot. At this point in time, this is the quality. Then you have the formulation, you have the behavior of the molecule, you put it on long-term exposure, and you can get different results. So you have to think about that from the beginning to gain the experience over time. This takes some money, but I think in the end it shortens the timeline. When an HCLF is not really, it's not possible, or you need a higher dose, required from the clinicians, then you have to take a higher injection device with a higher injection volume. Here as a subcutaneous classics, the prefilled syringe in 1 ml or in 2.25 ml is a common technology on the market, and you could put them in the needle safety device as it is required. All these things are available on the market, and you have to put them together. Auto-injectors, are still coming in a larger syringe format. It could be used a little more flexible because you could use them not only for 2 ml or less than 2 ml, you can use it from 1 ml to 2 ml. You can take it up what is really, what are the requirements for the later device and what does really the patient need. When we're talking about a really higher dose application, up to 5 or 10 milliliters, that could be a little bit more complex, so that you have to keep in mind that you need more time to bring the volume into the skin, in the subcutaneous area. And here are some new devices or devices in development with a point-of-care solution. This means that you have a standard packaging component that you put it into the device, put it on the skin, and later on you have an injection. And or you have a prefilled solution, which is currently under development from different partners. And this could be an ipsodose, for instance, or some other things where the cartridges and the formulation is put in on a direct way and assembled. The administration of biological drugs, so you can see here, when we're talking about vials, you can see on the x-axis the risk, and on the y-axis the benefit for the patient. Vials have really a low risk. It's a common standard, but for patients it's really incredible to have a complex preparation step before injection. Prefilled syringes are, as mentioned before, common standard on the market, have a low risk. It's more a higher risk related to vials because you have more impurities in the syringe here, like silicon oil, tungsten, or whatever comes up, and what kind of technology you need. And more standard is auto-injectors and needle safety devices, platforms for that. You can dealing with a good risk. You have more effort in development related to combination product requirements and functionality requirements, human factor engineering, and the necessary one is really the interaction between the user or the patients later on. And what we think is when we're talking about that HCLF is not possible, but we have a good protein in place that really helps patients, we can talk about a large volume device in the end. Currently, many of them are under development. It is a high risk, but it's a good benefit for the patient later on. They can do it at home, they put it on the skin, and it could work smart. But in the future, I think we have to develop the devices to standard platforms, which can be a little bit flexible related to the requirements of the formulation. But I think that is the future, that we have to decrease the risk for such kind of new devices to have more benefit for patient related to good drugs that help patients' treatment. I think that was my last slide. I hope I give you a good overview about drug product development within Boehringer Ingelheim and how we deal with it from a holistic point of view. And I can answer some questions if you have. I think everything is clear. If not, I'm the chair. I have. Okay, there. Thank you.
So, Christian, thank you very much for this excellent presentation. You briefly mentioned at the end of your presentation that combination products require some more efforts. And could you elaborate a little bit further from your perspective? What are the critical success factors you do see here in development of combination products?
I think from my perspective, within Boehringer Ingelheim, for instance, we get really the final dose from medicine very late in the development phase. Yeah, it takes some time within or after clinical phase 3, for instance. Yeah, and when we are talking about a device which is needed for phase 3, for instance, or to provide a bridging study during phase 3, yeah, then it is really less time to integrate device changes, for instance, from 1 ml to 0.5 ml syringe into the development process from a device perspective, because you have to have the changes in place from the device, you have to do the design verification, yeah, you have to do all these documents and all these testing, and sometimes it could be very challenging when you get the information too late. And this means that you have a little bit more effort in the end to bring it on the market, yeah. Because the user interface has to be evaluated, and at this stage you have to integrate it to the device development, and this takes some time, yeah, and that could be critical.
Thank you.
So then maybe adding on to that question, so do you foresee in the future then, or do you think it makes more sense then obviously to think about implementing or using devices earlier in the life cycle management already, like especially for ones that are new in development? As you said, start with the end in mind, so you start maybe with a vial and a syringe, but then while you're developing the molecule and everything, so you think about the device already,
yeah,
even if it's far ahead.
Mm-hmm.
Do you think that makes sense? Because you just said that it's too late, you have too short time to accommodate in the device development the wishes of the formulation development. So then when you start a new formulation development, that you already think about the device early stage on.
Yeah, you have to think early stage on. I didn't get the question correctly, I think, yeah, but you're talking about life cycle and products in the market, and if you do a life cycle but you have to have it from the beginning in mind, what is the behavior of the molecule to find the right solution for the patient, yeah. And I think that is really needed.
Are you more relating to the flexibility?
Sorry,
Thank you. Are you relating to the flexibility of the device that can take, a very wide level of viscosity without having to change anything in the device, such as the components, or when you're mentioning future devices,
mm-hmm.
is this the type of device you're looking for, something that is, flexible in terms of volume and viscosity, so that because, well, surely the behavior of the molecule might change as you go along and you arrive at phase 3. So that means that probably the future device will need to accommodate different fill volume and different level of viscosity.
Yeah, you need the flexibility with the device as well. Yeah, different springs or whatever components you have here.
Without having to change the spring.
You're having to, yeah, okay.
Yeah.
Yeah, I think that from a mechanical perspective, that could be very challenging, that you have an increase in viscosity, and to cover all these ranges with one spring, for instance, yeah, in auto-injectors, that's really hard to say. Yeah, you have to take the flexibility related to change some parts on the easiest way.
Sure.
Yeah.
So this is the future.
I hope so.
Okay.
Because that auto-injectors are a common standard, yeah, and we know what the technology is, yeah, and we learn how we have to use the flexibility from a formulation perspective, but later on we need it for devices as well, to start the discussion during development what is really needed. Yeah. Good. Thank you very much.